Yes, welcome. I've already seen some people let us know who you are and where you're tuning in from, so in the chat, so please feel free to do that. But before we more officially begin, I just want to share a bit about myself, my heritage, and recognize the ancestral history of Canada. My name's Hannah McCurdy Adams, and I use she, her pronouns. I grew up in Ontario, exploring the rivers, lakes, and the Niagara Escarpment. I'm second to at least fifth generation settler Canadian with ancestors from England, Ireland, Scotland, and the Ukraine. I work for Wildlife Preservation Canada. I'm on the board of directors for CHS, and I'm one of the meetings and workshops co-chairs along with Pam. I want to pay my respects to many nations, including the Mississaugas of the Credit, Anishinaabeg, Huron-Wendat, Haudenosaunee Confederacy, and Métis nations, who were the first keepers and current residents of the land where I currently live. I'm in an area covered by the Dish with One Spoon Wampum Agreement and Treaty 13, close to what is now known as Toronto. I also want to express my gratitude to those who've guided me to see nature from a perspective that is different from my own. Today we're meeting virtually, coast to coast to coast across Canada, and I'd like to acknowledge the ancestral and unceded territories of all the Inuit, Métis, and First Nations people that call this land home. As herpetologists, our natural lands and the animals within them are so important to us. This recognition is a
Yeah.
renewal of our commitment as a society to listening to and cherishing the traditional knowledge of Indigenous peoples. I also want to welcome international participants. I've seen a couple people say they're from New York in the chat, so welcome, welcome. And any of the experience and heritage that they bring with them. And so with that, I'm going to pass it off to Andrea, who's going to chair our first session.
Good morning through afternoon, everybody, depending on where you're at. Our first speaker this morning slash afternoon is going to be Sara Ashpole with research about Biotype does not explain climbing and autumn spatial distribution of salamanders in the blue spotted complex. So I will mute myself and let you go ahead, Sara.
Great, thank you. So I definitely want to acknowledge my colleagues here. This is research that was done by undergraduate students at St. Lawrence University. And also we've had work with Jim Bogart on some of the DNA work. So in many ways, I'm just the nerd who helps push them along in my coursework. But what it comes down to is we saw some behaviors that we haven't found in the literature. And I don't know why I can't. There we go. Okay, so if you know much about the blue spotted salamander complex, it's got a lot of complicated hybridization. And they're cold tolerant, terrestrial. They're kind of mole salamanders, so they're typically underground. They're insectivorous. They enjoy the leaf litter and they use burrows. And what we found was students would go out and do their surveys for part of the course. And next thing you know, they started to see these clusters. And so they started doing some fairly intensive surveys. And this area here, which is across the water from our campus, they were able to detect, individuals. And what they saw was individuals wanting to be really close together, and they were up on top of things. So this behavior of perching was like on vegetation off the ground. And we hadn't ever seen this before. And also, we saw a number of individuals touching each other or being really in close contact with each other. And so, a lot of close kind of connections. I do have to say that when we started the surveys, they were just going out looking for anything. And then, as a few nights went by, they started to develop a stricter kind of survey methodology to actually see the clustering effect and start to measure the height from the ground. We also know that because of the complex, we thought there could be differences seen between the different groups. And so ultimately, we actually saw nothing. We saw no connections between who they are and what their behaviors are. We saw no full Jefferson and 55 full blue spots, and then some of the hybridized. So no connections there. And really, the reason for this presentation was if anybody had ever seen this type of perching behavior in blue spotted or other salamanders, because we can't find anything in the literature, or if they'd seen any sort of gregarious behavior outside of like the mass migration of, let's say, breeding or in-pond breeding behavior. And so I think I was pretty quick, but I thought I'd give an opportunity if people had questions to do that at this point, or even just thoughts. And I know there's some good herpetologists in the crowd, so expect something.
Yeah, we have a couple minutes for questions. If people have questions for Sarah Ashpole, if you want to either type your question in the chat or raise your hand using the raise hand function. Oh, Beck, do you want to go ahead?
Oh, really interesting findings. I was wondering, did you notice anything like weird with the climate and weather and such? Do you think it was something to do with that?
So we did record a number of like climate variables, and we also looked at substrate type of variables as well. And we definitely honed in our surveying on good weather conditions. And so I would say we haven't really been able to detect anything there. So yeah, don't really know. But I mean, I survey a lot for a lot of different species in all sorts of survey conditions, and I haven't seen this kind of perching behavior ever before. Like seeing a mole salamander like a foot off the ground in like greenery is weird. I mean, it wasn't one, it was like multiple. But yeah. Um, Thomas?
Hi, Sarah. Yeah, that's really neat. I've never really seen ambystoma, climbing up on the vegetation, but of course, Patrick Muldown down in Bat Lake in Algonquin Park has found that spotted salamanders sometimes climb into pitcher plants,
Mhm.
and that's pretty neat. And we've also seen some pretty impressive climbing ability in captive held ambystoma. Like they can scale, like a straight glass aquarium wall, several inches off the ground. But it's usually restricted to smaller individuals. The climbing ability seems to degrade as the animals get bigger and bigger. So I was wondering if you saw any relationship between the ones that were up off the ground and their snout to vent length, or if that's something you measured.
Actually, amazing you mentioned the climbing into the pitcher plants, because actually that's a good thing I could reference, because that is clearly climbing on top of vegetation.
Yeah.
And I can reach out with them as well to ask what type of behavior they saw. As for this idea of like size, we do have size information. And to be honest with you, I don't recall the outcome, but that's something to double check, and that's a really good one.
Yeah,
Um,
I think the size influences their ability to successfully navigate those elevated perches. So I think it's probably more frequent in the smaller size classes. But that would be interesting to see especially if you have good data on it.
Yeah, thank you very much. Um, I love this question from Dana Shock about parasite pathogen. Oh, I don't even know how to go down that route, but that is really good, and something I can look into for other species, because I think it's a good hypothesis. And then I think there was another one.
Yeah, I think we can take one more question.
Oh, Fred's got a good point about the mud puppies in proximity. That's a good one too.
Um, yeah. Is this perching during mating season? Or
No, it's in the
no?
fall. It's in the fall, and so like this is October, November, like September, October, November. Um, and so it's definitely not during breeding. Yeah, good.
Okay,
Um,
cool.
Histopathology, if they were dead, I guess, right? Maybe. That's Dana. Okay, thanks,
Um,
anybody.
all
Anybody can
right.
later.
Yeah. Thanks, Sarah. Sorry to cut you off, but this is we're keep so I'll thank you, Sarah. And I'll call on our next speaker, Jordan Voss. If you're
Hi.
here. Hi, there you are. Okay, Jordan Voss is going to give us a presentation, diet analysis of prairie amphibians from the University of Lethbridge, Alberta. And you can go ahead, Jordan.
So my name is Jordan. You can also call me Joe. I'm a master's student at the University of Lethbridge, and my topic is diet analysis of prairie amphibians. As a little bit of background, this is kind of what I'm hoping to cover today. It is very rudimentary, and I will explain why. So as I mentioned, my name is Jordan, or Joe. I am a professional frog nerd, and I graduated with a Bachelor of Ecosystem Management from Lethbridge College. I now actually also teach at Lethbridge College and absolutely love it. I am working on my master's at the University of Lethbridge under Dan Johnson, whose focus isn't primarily amphibians, but it is insects, which ties nicely, especially with the topic that I have. I love taking anybody I can out with me into the field to teach them more. This is my fiance, who I drag out for most of my field studies, so we have a lot of fun. My passion started when I was really young. I have experience with almost all the amphibians in Alberta except for the long-toed salamanders. I've worked as a park interpreter. I've shared my joy for passion for amphibians and stewardship through a lot of different opportunities that I've had given to me, and I also share it with a lot of younger generation, including my siblings. So I love amphibians and wanted to study them for a very long time, and never thought that my career would take me where it has now, but super grateful for it. So for the background, how did I get here? Originally, my master's topic, I was hoping to study the effects of pesticides on amphibians, particularly the boreal chorus frogs in southern Alberta, because they are very common, and they are commonly in areas that are affected by pesticides in agricultural cropland. And I had many different questions, and there are very few answers. From what I did in my initial literature review, I found that most of the studies for pesticides and amphibians relied on data that was for the tadpole or larval stage and very little on the adult phase, which didn't make a lot of sense to me, in the sense that if you're taking up oxygen and moisture through your skin, you're likely to become more inundated with pesticides as you encounter it in the landscape. So I had many, many questions and very few answers. I originally also wanted to look at the effect of pesticides being taken up through the diet of boreal chorus frogs, and there was very little knowledge or publishings available about what they were specifically eating and whether or not those insect species were actually affected by pesticides. And of course, we know amphibians to be one of the most at-risk groups of animals. So it's very important that we consider these different aspects. And I find it really important to consider this because of their overlaps and their habitats between their life cycle. And it feeds really well into how we eventually form conservation practices. My study area focuses primarily on the Silver Sage Alberta Conservation Association site, which is in the southern area, a little bit to the southwest of the and I'm hoping to add a few more sites this season. This is one of the larger wetlands that are on the site, and there are calling boreal chorus frogs in this area, as well as possible other includes plain spadefoot toad, Great Plains toad, tiger salamander, as I mentioned, boreal chorus frog, and possibly the northern leopard frog. This site is not affected by pesticides, so it does feed in nicely to actually studying some of their native-ish habitat. It is a bit of a reclaimed native prairie, so it's interesting to compare those habitat sites as well. And this is one of the studies that actually discusses the restoration of the mixed grass prairie. So it's really fascinating, and Dan Johnson, my supervisor, has noticed a very obvious difference between the grasshopper species, particularly between the recovered grasslands and the native grasslands, as well as just the modified grasslands and the different species that are available. So it's really interesting, and I'm really curious to see how that plays out in the diet composition. This is my supervisor out collecting insects. He also shared with me a couple other images from the site as well, including some of these. So this is one of the modified areas of grassland. Some of the reclaimed that is growing back. So my objectives for my study, I wanted to implement effective capture and survey techniques, so I'm using pitfall traps. I also want to use stomach flushing for diet analysis, so I'd love to open up a discussion at the end if anyone has experience using this particular method, because from what I've seen in studies, or at least what's published, I don't see many people using this, especially in Alberta. I want to identify diversity, explore the relationship between amphibian morphology and prey size, and compare the habitat types of insects. So pitfall traps, drift fences. These are slides that I usually include for a more broad audience, just because most people aren't familiar with these, but I'm sure many of you are. I also found out last summer that mix well with cattle. They did not let me know that they were putting cattle on the site when they were out. So I know that it is a grazing area. I just thought that I had a little bit more time. So the image on the left, set up, good to go. Image on the right, kind of questionable. But we did manage to catch one boreal chorus frog young of the year. And I've had many kind of pitfalls in my own research. I'm technically into the kind of last semester of my master's. I'm extending it because I've had a lot of things happen both in my personal life and with the study itself that have kind of pushed my timelines back a little bit. So I don't have any data, warning you now. So this one little boreal chorus frog, I actually caught 3. One escaped, and the other 2 I brought back to the university so that I could receive my stomach flushing training. And that's kind of this. So from the literature review that I did, I found that there were 3 main methods for the dietary analysis, including stomach dissection, fecal and DNA analysis, and stomach flushing. And of course, each has their pros and cons. I found that stomach dissection was commonly used with invasive bullfrogs. Fecal and DNA analysis is kind of getting more common. The drawback being you have to hold them a little bit longer until they expel their fecal matter. And as well, if you're performing your DNA analysis, you're kind of missing some of the pieces if you don't necessarily have the proper primers to see all the things in their stomach. So that's where stomach flushing kind of came in my study and why I found it to be beneficial, because it's very quick, it has a short turnaround time, and it doesn't require any anesthetic. Now, we haven't gotten to lunchtime yet, so we hopefully shouldn't be losing our appetites. But this was me performing the stomach flushing on this particular individual. I did feed it 2 fruit flies just to see if it would be effective. And I know that the image is a little bit grainy and it's hard to see, but those are the full 2 fruit flies, which shows that it is fairly effective. And I'm hoping that I get really good information from this. I also do geographic information systems analysis because I teach GIS at the college, and I'm hoping to perform some analysis on the diet and items and habitat ranges. So I'm planning to use GIS to determine what prey items might be selected by each species, body physiology, and how their body types influence what prey they eat, the timing of prey item emergence, their size of the prey, and the themselves. I have read in some studies that the gape size of their mouth will actually influence what they eat. Obviously, you're not going to eat something that doesn't fit. I'm also interested in the prey capture methods between whether they are active hunters or if they're more. And I'm also interested in how climate change and the occurrence of drought impacts these species. I find it fascinating how the plain spadefoot toad is particularly different, but I'm curious how other species also react and how that influences the diet as well. So just as a bit of overview from the GIS side of things, so these are some of the insect traps. These were insect sites that we collected data from. And these are some of the different vegetation types that are on the area. And hopefully you can see my mouse. This wetland in the bottom left corner here is that big one that was in the image, and where I have so far set up pitfall traps, and I'm hoping that this coming field season is going to be a very productive one so that I can make it out to a few of these other ones. But there is a lot of different vegetation types. So there is the native grassland and grassland reseeds. So these are the reclaimed areas. I will note that this kind of tame pasture area that is right close to the wetland is mostly crested wheatgrass and is extremely modified. So I'm really fascinated to see how these different habitat types will actually influence both the amphibians and their diet species. These are just an image of the insect pitfall traps and some of the species that are found on the site. Now, my grasshopper identification is not nearly as good as my supervisor's, but these are some photos that he provided to me, and I'm interested to see if the sizing and timing and different life stages will actually influence the species that are being eaten. As, again, I don't have any official results yet. My current hypotheses are that stomach flushing will provide a detailed and accurate assessment and hopefully give a little bit more input and information to compared to the traditional methods, dissection and the fecal analysis. I'm also hypothesizing that prairie amphibians will exhibit a diverse range of dietary preferences. So showing will adapt to different ecological niches and variations among the species, as well as the size and morphological adaptation, that there would be a correlation between the size and the features of both the amphibians and the prey items that they consume, show that they adapt to their feeding behavior. And I also hypothesize that different habitat types, even at small scales, will influence the diversity and abundance of insects, and therefore the variations will correlate with the types of prey that we amphibian stomachs. And of course, has a lot of significance. I'm really, really excited to get input from other herpetologists today, because I don't have a lot of people that are within my field that actually can help me work out some of the kinks. So I would love to have some feedback going into this field season. I'm particularly interested in the significance because diet items lead into food chain protection, other implications. Like I mentioned originally with my research in pesticides, I'm really fascinated to see where this ground level information will actually feed into other studies. As well, eventually, if this turns into a multi-year project, I'm interested in the nutritional value and how it feeds into overwintering, especially in species like the northern leopard frog. Also, intrinsic value. I'm, again, super huge frog nerd and love to share that with others and really want other generations to see this in the future. Even as a kid, I've recognized that the populations aren't what they used to be. I remember hearing and seeing many amphibians as a kid, and now it's just feeling like there's not as many left. As well as the conservation and productivity of rangelands. So I am a farmer and rancher's daughter, and I recognize that when you have amphibians on your grazing lands, you're likely to have better productivity. And whether or not they're actually linked in the sense that if you have amphibians, you're going to have good grazing land, it's probably more of a tied situation where if you're actually preserving the land, you're not only preserving it for the organisms that live there, but also for the productivity of your own business agriculture. So I really want to strongly advocate for that. And I also want to recognize factors that lead to the presence and absence of amphibians. So not only pesticides, but also habitat change and other parameters that lead to their demise. I'd love to protect them far into the future. These are a whole bunch of my resources, and I still have more to add. But I will open it up for any questions, and I will just leave this slide up for a little bit. So if you have questions after the fact and want to, or information and feedback, you can also email me at my email there, or you can connect with me on LinkedIn. I don't check it very often, but I do try to once in a while. So yeah, super excited to hear from you guys.
Oh, thanks, Jordan. Yeah, we've got time for a few questions. If people have either want to
Actually, Jordan, I have a quick question, and then I'll let Andrea deal with Fred's in the chat. Question/comment, be really interesting. One of the things I'm doing right now is some climate change modeling, looking at different species and how they're going to react to things. So that's a little piece of your puzzle, but I'd be happy to connect and share the stuff that I'm doing. I'm in Manitoba, so I'm also, yeah, a fellow prairie folk.
Awesome, thank you. Looking forward to it.
Okay, from the chat, Fred says, look at my Burn Henry Ford and Effigy article in the Canadian Herpetologist. Seems that the leopards go to the hibernacula later on in droughty summers when there hasn't been lots of summer food. So that might be something to look at. And then we have another question from the chat. Do you have databases / methodology in place for insect area analysis in GIS?
So I don't have any currently, like I don't have any open source, and there is very little from other sources. So this is kind of something that I'm developing with my supervisor from the ground up, particularly because he is one of the leading experts in a lot of things insect world. So it's really nice to have him as part of my team and my supervisory committee. We are working on getting it a little bit more updated in a GIS format, especially because he has a lot of historical data that isn't digitized. So it is a very large undertaking trying to bring that into a GIS component so that we can actually perform different things on it. I do have some. So from that one map, do you still see my screen? Okay, I'm going to just quickly go back to my one slide here with my map that I created. So I do have some of these points mapped out. He had, I think, carrion beetles, spiders, and one other that off the top of my head that I can't quite remember. I did do some very preliminary brief analysis to kind of see where different species would show up in greater populations. So that is kind of where things are at this very moment. And I'm still trying to develop a few more things just to make it a little bit more and to make it actually more applicable. The other thing that is interesting is the difference in species between his surveys and when he uses his nets versus when we use the insect pitfalls, because some of the grasshopper species are both extremely tiny and also don't have wings, which would likely feed in very, very well to an amphibian diet, especially being on the ground and your only escape being hopping away. I mean, if your predator is also hopping, it kind of levels the playing field a little bit. So I'm really, really interested in how that all plays together. And I'm really hoping that it turns into a really good GIS project. This will likely turn into a doctorate situation, but it's just been a lot of things that have led to me to where I am now. So it's both a win and a little bit of a lose. I feel like I've lost a lot of field seasons just with everything that's happened. For some minor backstory, my first supervisor actually retired on me 2 months in, which left me in a limbo state for like half a year. So it was extremely frustrating, and I'm trying so hard to play catch up now, which is why it's taking a little bit longer. Yeah.
Great. Thank you, Jordan. And we'll just we'll move on to our next speaker just to kind of keep on time.
Awesome.
But really great talk. Appreciate that. Our next speaker is Haley Grieve from Trent, who is going to be giving us telling us about factors affecting activity range selection in sympatric eastern red-backed and blue-spotted salamanders. So
as I said, my name's Haley. I'm from Trent out in Peterborough, Ontario. And my supervisor is Thomas Hosey. My secondary reader is David Beresford. And today I'll be presenting the factors affecting the red-backed and blue-spotted salamander activity range selection. So these are my two study species, the eastern red-backed salamander and the blue-spot Ambystoma salamander, which I'll be referring to henceforth throughout this study as just Ambystoma. So Ambystoma have some key differences. They get a little bit longer, they have a larger mean activity range, and they're known to displace further than their red-backed counterparts. However, both these species are labeled as generalists in range and diet. So if their ranges and food preferences overlap, then how do these two species coexist? Do these species partition their niches for dietary differences, or do they occupy different activity ranges due to different prey availability? My research question asks, what factors affect the selection and overall area of an adult salamander's nocturnal activity range? I have two hypotheses, the first of which says that the prey available within these activity ranges affects where both species choose to forage, with the prediction that there'll be more prey available within their ranges opposed to paired random sites. And my second hypothesis states that salamander activity range is affected by their snout to vent length and species, with the predictions that larger snout to vent length will result in larger range size, and Ambystoma will have larger ranges than redbacks because they're large in general. So I collected my data from September through October, and during this time period, I would go out and collect my salamander and habitat characteristics, as well as their activity range sizes, and the prey availability within these ranges and their paired randoms. And then into December, or then collectively, I would extract the prey from it using Burley's funnels, and I did the prey ID into December. This resulted in 39 data points for each species, resulting in a data set of 78 salamanders. And I collected all of this data at the Trent Oliver Center, located about 10 minutes outside of Bob Cajun. So for collecting salamanders, I would go and find them. I would collect their posterior snout to vent length and their mass. Then I would get the soil temperature and moisture of their cover objects. And then I would put them in individual bags of powder and then coat them gently and release them back under their original cover objects. I would repeat this for 9 salamanders per night. And then the following morning, I could come back and actually see where they traveled overnight because they would leave trail markings for where they foraged. So this is our demo range. So the purple here represents their activity range trail. So to get the area, I would first take their longest length traveled, multiplied by their widest width to give me an estimate of sinuosity, and then I could use those 4 points and measure the lengths in between them, giving me an overall total activity range polygon for that salamander. So within these ranges, I would then collect the soil moisture and temperature, collect 1 meter squared of leaf litter from that centralized point, and then 0.001 meters cubed of a soil core volume, which I would then use for prey availability. I would also walk from that starting site, I would walk 5 meters within a random coordinate direction, and that would be the paired random site. And I would repeat this over there as well. And I would do this for all 9 salamanders that night. Then I would take these leaf and soil samples and put them into Burley's funnels, layered soil, then leaves, let them sit for 5 days so the invertebrates could escape the heat and crawl out the bottom into soapy cups of water where they would drown. I would then strain out all the invertebrates and then freeze them individually for later prey ID to order level. So from our prey analysis, we focus our prey availability analysis specifically on taxonomic groups that are known to be consumed by salamanders of these species in pre-existing published literature, and also that occurred in at least 10% of the occupied activity ranges for both Amphibia and redbacks. So for my analysis, I first conducted a conditional logistic regression model with habitat characteristics as the predictors for each species to evaluate the differences between their activity ranges and paired random site characteristics. I then formed data sets with the most frequent prey compositions for each species, then conducted a principal component analysis to further reduce those compared prey types so that I could then put them into a conditional logistic regression and test for significance of prey in their ranges versus paired randoms. And I then did the same analysis, but comparing between species within the prey in their both activity ranges. So no randoms for that one. And finally, I used a generalized linear model with a negative binomial error distribution to compare each species' activity ranges with their posterior snout to vent length, and I also used rain as a covariate. So results. I found that canopy cover, soil temp, moisture, and leaf mass were not significant at the 5 meter scale for either species. And then from my PCAs and conditional logistic regression models, I found that when comparing species, there were no significant differences. When comparing redbacks to their paired random, also no significant. But Amphibia did have a significant conditional logistic regression, meaning, as you can see here, it is significantly negatively correlated in their occupied ranges opposed to their unoccupied ranges. So essentially, everything that's positively correlated here, they want less because, again, negative significant correlation. So Amphibia are selecting sites that have less millipedes, earthworms, non-orbitoid mites, springtails, fly larvae, and selecting sites with more beetle larvae, orbitoid acari, and orbitoid acari. So I then found that when I tested for differences in activity range size between species, redbacks had less large ranges than Amphibia. Amphibia snout to vent length was positively correlated with their ranges, and that rain was a negatively correlated factor for the movement of both species. So now discussion. None of these 4 habitat characteristics were preferred compared to their paired random sites, which means that foraging selection is likely occurring at above the 5 meter paired random scale that I used. Other possible factors could be the moisture of the leaves as opposed to their dry leaf mass. Maybe they're looking for a moisture level as opposed to how many leaves they have in their ranges. Or perhaps proximity to water, maybe the vegetation composition, they have a preferred type of plant. Or maybe terrain conditions such as elevation could be affecting their movement. They could be choosing the path of least resistance and the most depressed angle instead. There is support that Amphibia salamanders may be partitioning their niches to have more beetle larvae and orbitoid acari mites. So this could mean maybe there's dietary constraints, so maybe other more competitive species are forced, like the territorial redback salamanders in the area, are forcing them to specialize to certain prey types. Or perhaps this allows for the unisexual complex to continue existing. Alternatively, it might just be because of abundance. As you can see in the prey sum chart to the left, orbitoid acari are by far the most abundant prey type out of any I collected. So maybe it's just a matter of it's here, I'm going to eat it, and it's consistent. So for redback prey analyses, again, we found no differences between species or between redbacks and their randoms. This might indicate that prey is overabundant for redbacks and they don't really have to specialize like their Ambystoma counterparts do. Or maybe this selection happens at the above 5-meter scale. Alternatively, since I sampled only in the fall, maybe this seasonally differs in their breeding season in the spring. Maybe the prey that they're selecting for, they may have a dietary niche for when the males are establishing their territories during their breeding season. Or perhaps they're foraging elsewhere. During our surveys, we found that during especially rainy nights, redbacks were very likely to forage on trees. So maybe this could be negatively impacting their range size, so they're foraging in vertical space opposed to overall activity range. And also rain negatively correlated with both species, likely because the powder just washed off a lot easier with the increased moisture in the environment. So for snout to vent length, we found that larger Ambystoma snout vent length resulted in larger ranges, but we did not find the same relationship for redbacks. This may be because we have a more drastic size range, as you can see in the blue highlighted text. We did capture some juvenile Ambystoma and some much larger adults than any adult redbacks that we captured. This might be creating a more drastic size range that shows a trend in the data as opposed to not just in the redbacks. So these results might also show for redbacks if we did sample some juveniles as well. Alternatively, foraging might be determined again by elevation for redbacks. As you can see, they were climbing trees, and this may also vary seasonally, again, breeding season. We also found that Ambystoma ranges were generally larger than redback ranges, or significantly larger. This might indicate that foraging styles and frequencies may differ between species. We found that Ambystoma frequently stayed burrowed when there was nights of extreme temperature drops. They were more likely to just stay nice and snug in their cover objects, and they did not venture out. However, the redbacks frequently did go out, and even when it did drop colder, they would just kind of go shorter foraging ranges as opposed to staying hunkered down. However, when Ambystoma did choose to go forage, they would forage longer than the redbacks did on average. And another key point is that redback salamanders had the 2 largest and most directional foraging ranges that I collected out of the entire data set. So future studies should think to compare between species-specific foraging patterns and frequencies. If maybe Ambystoma are more likely to do these centralized foraging tactics where they find a prey and they just keep going until they have their bellies full. Whereas redbacks, they could go up to 13 meters in one night. The largest range I ever had for an Ambystoma was like 7 meters tops. In conclusion, redback range size is not affected by snout to vent length or prey availability, and this supports a generalist feeding strategy for the species at least a 5-meter scale, and we also noticed that they do forage in trees on nights of increased rain and moisture. We found that Ambystoma ranges were significantly larger than redback ranges, and Ambystoma foraging is longer when they have a longer snout to vent length, and there is support for dietary niche partitioning, and they favor sites that have beetle larvae and orbitid mice. We also found that UV fluorescent powder can't be reliably used in conditions with intense rain or frequent rain, lest the trails wash off or the powder gets brushed away a lot quicker. And we suggest that future studies should explore foraging frequencies and patterns between individuals and species, so that can give us a better indication of the movement ecology on the individual variation level as well as the between species level. Thank you for your time.
Oh, cool. Great. Thanks, Haley. We can take probably 2 questions if people want to. Oh, this one right away. Okay. Michelle asks, what did you use for the powder? Obviously, this wouldn't negatively affect them. Great presentation.
Yeah, totally. No, I love these guys so much. I would never want to harm a salamander ever. So we had quite a few different papers that we were able to cite preemptively for our animal care protocols. I don't know the exact brand of UV fluorescent powder, but if you want, I can go and find that paper and send it to you after the presentation. And it shows essentially that when they're coated in it, it doesn't affect their respiration, their vision, or their foraging. So by all means, it's just a means of where did they go and how far did they go.
Jordan, can the powder be used on other amphibians?
Yes, actually, there have been a few other studies that use it on frogs and toads, and it's really cool because you just see their little it's like spots that they leave as opposed to the consistent salamander trail. So yeah, it has been used for other herps too.
I think we can take one more. If anybody has a burning question?
I'll just jump in and say, Haley, you're getting a lot of generally the salamander stuff is adorable and cool.
Yeah,
The fluorescent powders in the chat.
it was a really neat project, and I'm really grateful that I can be here to share it with you guys.
Okay, well, we'll keep on schedule. Thank you, Haley. That was great. And we will welcome back Sara Ashpole from St. Lawrence University. And we're gonna learn about the Amphibian Specialist Group, status and trends of the world's amphibians and Canadian Amphibian Conservation Action Plan priorities.
Awesome. Thank you very much. So in many ways, this presentation is really just to highlight some new items in the literature that are really good resources for people versus me going through the documents. And I'd like to say that a lot of this work, both Christina and I myself have been involved with tangentially and in some cases more directly. So definitely this is kind of useful information. But in case any of you are just curious, the Amphibian Specialist Group is part of the IUCN Amphibian Specialist Group here in Canada. And so we focus on the Canadian perspective, but we report back on the global scale. And we renew every single 4 years. And it actually lines up with the World Congress of Herpetology, which is in Kuching, Borneo this year. I really hope a whole pile of you are going because it's extraordinary. And I could give you a lot of other bits of detail, but if you're interested in joining as an expert and there isn't even a student level, just reach out to me, okay? But definitely know that the website has got a lot of materials on there that individuals can access. If you're thinking a little bit about what do we do here in Canada, we're interested in making sure that we're understanding and reporting on declines and keeping up-to-date information on diversity and changes in Canada. We have some action items around, protection and management and restoration for Canadian species. We do a lot of collaboration. CHS is a really good example of that. Partners in Amphibian Reptile Conservation is a good example of that too. And we like to promote a lot of mentorship and initiatives to create networking. So sometimes we get an email saying, hey, I'm looking for this. And we just try to find you the right people to connect to so that we have a bigger expert network and what have you. And it goes across borders too. So the first document I wanted to share with you was just recently published in Nature. And this has 73 authors. These are the co-chairs, global co-chairs for ASG. And it is a status review. So it looked at the red list at over 8,000 species. And lo and behold, amphibians are the most threatened vertebrate class. And we kind of knew that before, but they remain that. So we haven't really abated the extinction risk, even with what might be seen as quite a few newer, more recent activities. Disease and habitat loss, not surprisingly, drove 91% of the deteriorations for a long period of time, up until about 2004. But more recently, since then, 39% has been driven by climate change, and then 37% by habitat loss. So definitely one thing to think about is the most significant improvement in protection was actually done through habitat, providing habitat measures, except we actually know that a lot of habitat measures that are on protected lands, that they're still sufficient because there's not enough policy around protected lands either. Okay, so not surprisingly, need for stronger policy, political will, and sufficient resource commitments. This is a great article for people to be referencing with up-to-date information. So there's that article. The next one I wanted to point out is something that emerged from that is a really highly user-friendly graphical state of the world's amphibians. I just gave you some examples here. This document is about 30 odd pages and is just tons of visuals. And these visuals you can cut and paste and put them into like lectures, into like outreach activities you're doing. Phenomenal summary of information in a really visually pleasing way. So definitely keep your eye on this when you're thinking about educating and outreach and what have you, because it's so nice. And then I wanted to point you all to a almost 800-page tome of information, which is called the Amphibian Conservation Action Plan. And this is the third one, and the third one is actually done quite differently than the first two. So I've given you the link to where you can access this document. It's still in the final draft phases, so it's not fully formalized, but it covers, every main thematic concept. So there's 14 chapters, and they're kind of divided into threats and decisions and species. And they identify things like what has happened in the last 15 years. So really recent up-to-date information, looked at data gaps, looking at how to think about, take-home messages for people to use it. The nice thing is there's going to actually be a follow-up document that's going to be very short, so they say. And the short document is actually supposed to be a public practitioner document. So this is for the wider audience on recommendations. And when you were all sitting there going, oh, should I join IUCN ASG Canada? This is where people who are really into making sure that recommendations are out there, you can participate in the writing, but you can also participate in the review of this type of information. Okay, and if you're all wondering like, well, we should be doing something in Canada, there's a whole team of us that are actually putting together a research paper, and we're probably about 3/4 of the way through the process. And this involves looking at the full ACAP, but from a Canadian perspective, and looking at those kind of challenges that we have in Canada with like northern distribution limit ranges and conservation. I want to say I'm really proud that our document for Canadian ACAP actually has an Indigenous writer on the team, and putting that perspective in is something that doesn't exist in the global ACAP. And also we're trying to cover off topics that haven't been covered, which includes ecological restoration and work on non-native invasive species. So that's looking really cool. And pretty much it was really just an information sharing, but I'm hoping some of you are interested in joining ASG. We really rely on experts for participating, but also networking. And with any questions, I guess that's everything. Thank you.